Hi, hello everyone. Thank you so much for welcoming me here. This is my first time in Hong Kong and Macau, so I've been just really enjoying the warm welcome. I also want to thank all of you for coming to listen to me today. I'm really excited to share about the work that we're doing, mostly in the United States, but also I know it's a weekend and you're devoting your time to be here to listen to my ideas and what I think is important, so thank you so much for your time and attention. So, a lot of my work really thinks about the kinds of environmental influences that shape child development. And we know that children can encounter many different adversities as they grow up. Now, these adversities vary in type, kind, extremity. They have different features. But we know that in general, early experiences of adversity have really profound effects on the developing child. We know that early adversities tend to travel with a child throughout their lifespan and are very privileged in their timing, right? Early adversities matter much more for a child's development than an adversity that's encountered later in life. So luckily, Dr. Wong really set this up nicely for me, so I don't have to spend too much time talking about theory, but I do want to just remind all of us together of two really important central ideas of child development and developmental interventions. One of these ideas is the idea that early experiences matter more. Things that happen early in life have an outsized impact on later outcomes in comparison to events that happen later in life. And we see this not only from this figure that has already been explained to you, but we see this in humans, we see this in rodents, we see this in monkeys. This is not just a thing that's specific to humans, it's true across a variety of species and in a variety of different adversities. Second, we know that there are sort of sensitive periods in development where certain experiences have a much larger impact on how our brain develops. So there are times where our brains and our bodies are ready for specific things to happen. And if those experiences happen in those times of sensitivity, they have incredible cascading effects for the rest of development. So there are times where our brains are ready for specific experiences, and we've evolved to have this be true. There are times that our brains are ready for social interaction. There are times where our brains are ready for taking in of light and organizing our entire visual system. There are times where our brains are ready for taking in sounds and organizing our auditory systems. And as long as those experiences happen, our brain organizes in a way to support expertise and development. But if those experiences don't happen in those sensitive windows, it can have everlasting consequences for how our brain is organized and how our behavior unfolds. So, I just realized this is showing me one behind, I'm sorry. So, if we want to think about how to help children achieve their optimal potential, how we make children be the very best they can be, given everything they walk into the world with, we really need to think about how do we shape the early experiences a child has, but also we need to understand how early adversities, when not preventable, impact developing systems and the developing brain. In particular, one of the things we want to think about when we consider intervention science is we want to think about how do we identify early mechanisms or markers that are shaped by these adverse experiences. So we've seen many present markers and many measurements today that sort of show us early predictive markers of later behavior. And particularly when we think about identifying mechanisms or markers that are related to early experiences or adversity, we want to identify mechanisms and markers that are usually showing us when development is going differently before we have an outcome of interest, right? So for example, if we want to think about how do you help a child have great language before they enter school? Well, we want to start measuring how is language at 1 month old, 1 month old, how is language at 6 month olds, how is language at 12 months old, and start understanding where you can start seeing deviations in development so that we can start intervening at times that are earlier and at times where the brain is very sensitive to those interventions. But we also want to identify markers of development that are sensitive to intervention, right? If we find a marker that is telling us, okay, language development is off course, or socio-emotional development is off course, there are early signals that this might not go well. If that marker isn't malleable to intervention, we can use it for identification, but it doesn't give us an idea of are interventions working? Are our efforts to put development back on a different trajectory working? And so this is something that we want to think about when we think about combating the effects of early adversity on the developing child. Then, of course, on top of identifying mechanisms, we always want to think about trying to drill down or add specificity, have finer grain understandings. You know, we might get understanding at a global brain level, but then what does that mean at a cellular level? What does it mean? What are the mediators and moderators? I'll talk a little bit less about this today, because today I'm really going to talk about these sort of first 3 steps of this model. And today I'm going to spend some time talking to you about work that I've been doing, but a bunch of my colleagues have also been doing, thinking about an early proposed marker of early adversity, and one that we think might be really helpful for understanding the effectiveness of interventions really early in life. And that marker is functional brain activity, particularly resting functional brain activity. So in order to tell you about this marker, I think it's important that I give you a little bit of theoretical background, because I understand that some of you come from all sorts of different backgrounds. So I understand doctors and nurses and speech pathologists, and I don't know how much background you have in resting EEG. So let me just tell you a little bit about brain activity development. So if you're not familiar with EEG, it looks something like this. You can see me here with my own daughter, who's still in her first 1,000 days of life right now, and at home with her dad right now. But here you can see my daughter is wearing an EEG cap, and the second child here in this picture is also wearing an EEG cap. Now, some EEG caps look a little bit different than these ones I'm showing you here. Some are more mobile, some are more portable. In fact, now EEG technology is getting so good that we have little adhesive stickers you can stick onto the forehead, and you can take them into the field and into homes, and that's something I've spent some time doing in my work. Although I will say the signal is not nearly as robust to movement as FNIRS. So why use EEG? Well, EEG is really relatively cheap in the neuroscience world in comparison to fMRI or FNIRS. Well, we always love to talk about the fact that we're much easier to fund. It's a much cheaper technology. But also, it's really not very invasive. The great thing about EEG is it's just measuring the electrical activity of the brain. So when we think about measuring EEG, it's what we call passive. We don't send anything into the brain. We only measure what's coming out of the brain. And we usually tell parents it's kind of like how my voice is going into this microphone, right? My body is putting out sound waves, and then this microphone is picking up those sound waves and measuring them. Sort of the same thing, but it's in a hat form, and we're measuring brain waves. When we measure brain waves, we get what we call, oh, you can't see my mouse. Hold on. Let's see if I can get this going. Can you see it now? Great. So we get this, what we call a complex waveform, but you could also just call it a very squiggly line. And what we do with this very squiggly line is we break it down into what we call oscillations. And we have, for the not great for science communication, we name these oscillations by Greek letters. So we have delta, theta, alpha, beta, gamma. I'm not going to try to make you memorize all of those today because it's a sort of a working memory problem. You have to keep all of these bands in your head all at once. It's a lot. But what I do want you to understand about each of these frequency bands is that they oscillate at different rates. So some are slower. So here you can see theta that's going up and down more slowly, and some oscillate faster. So things like gamma up here that oscillates much more quickly. And the speed of the oscillation, we think, has a lot to do with the kind of information that it carries and the sort of information it carries in the brain. Each of these bands have their own functional definitions. They're good for certain things. They have different definitions at rest or when they're evoked in a task. And I'm not going to try to get you to memorize all of those today, because what I want to talk about is really just the general developmental pattern of EEG. So for today, I'm going to split these frequency bands into two categories, one of which I'm going to call lower frequency brain activity, and the other of which I'm going to call higher frequency brain activity. So higher frequency brain activity oscillates faster, lower frequency brain activity oscillates slower. And so here I'm going to show you about what typical development in EEG looks like. So here on the x-axis of this graph, we have age in months. So we have 5 months here and 51 months here. And on the y-axis, we have percentage of brain activity. So you can just think about it as a percentage, what percent of your brain activity is in this specific band. So first, let's talk about lower frequency activity. As children develop, we see that lower frequency activity decreases. So across development, lower frequency activity decreases. In contrast, higher frequency activity increases. This is just the typical development pattern. So now let's move this typical developmental pattern into the adversity world. What happens when a child has an experience of early adversity? Well, to understand this, I think we should just talk a little bit about why we would care about adversity and how adversity would shape the brain. We know that brain development is the most rapid in the first few years of life. And this is true in sort of two different ways. One way is that we're born with a bunch of brain cells, but over the first 1,000 days and beyond, we know that the number of brain cells we have increases really rapidly, particularly in the first few months of life. And then usually we prune some of those brain cells away as well. But it's not just the number of brain cells, but it's also the complexity of the connections between brain cells. So here I'm showing you some brain cells here and their connections. And as you can see, even just across the first 2 years of life, the complexity and number of connections between these brain cells also increases. And because this period is such a rapid time for development, we know that experience shapes and impacts both of these processes, the number of brain cells and the complexity of brain cells. So this is why we think measuring brain activity might be of particular interest and might be something that's very sensitive to early experiences of adversity. So when we think about adversity and how it impacts brain development, we have this hypothesis that, in typical development, we see that lower frequency power is decreasing and faster, higher frequency power is increasing. And we think that adversity is likely to affect this transition, that children might not show the same expected developmental change in lower and high frequency power. So is that true? Let's see. So do we see evidence for this change in patterns of adversity? So here, the first study I'm going to show you is by a rather famous study at this point from a number of years ago. And in this study, these authors looked at children who were being raised in institutional care in Bucharest, Romania. So just to give you sort of the context, the idea of a quote-unquote orphanage, but most of these children actually are not orphans by any stretch. In the imagination, they have living parents, many of which have not terminated their parental rights, but they're being raised in an institutional setting. And this research team went in and they looked at the brain activity for children who were experiencing institutional care versus the children who were not experiencing institutional care in the community. And for the lower frequency brain activity, I'm going to show you what we call a topographic heat map in a second, but I want to show you, sort of orient you, because I'm going to show you a bunch of slides like this. So here what we're doing is we're looking at the head sort of from the top down. So if you think about like if you were above me and looking down, this little triangle is the nose. And our hypothesis is that early experiences of adversity will result in more lower frequency power because that lower frequency power isn't decreasing as we might expect. So if that hypothesis bore out, you would expect to see warmer colors over here for this lower frequency slide and cooler colors over here. And that's exactly what these authors saw. So they saw in this lower frequency band, this trade-off wasn't happening at the rate that they expected as a result of early experiences of adversity. But what about the higher frequency band? Well, here we would expect to see the exact opposite pattern. We would expect to see cooler colors over here and warmer colors over here. And that's exactly what these authors saw here. So this is some early indication that early experiences of adversity are shaping how the brain is developing in this functional brain activity measure. Do we see this pattern in other experiences of adversity? The answer is really yes. So here is another map. You can see there are differences in warmer and cooler colors again. And this time I'm showing you data from children who have experienced poverty and lower socioeconomic status. The interesting thing about poverty and low socioeconomic status, though, is that we don't see this relationship when children are born. So this gives us an idea that, again, we see this difference in power trade-off in lower and higher frequencies, but this pattern really becomes stronger as children have more experiences over their first year of life. But by the middle to the end of the first year of life, we see strong associations between early experience of adversity, poverty, and brain activity. In my own work, I've spent a lot of time thinking about stress as a particular mechanism for how this pattern may unfold. And so we know that children who are being raised in poverty, but we also assume that children who are being raised in institutional care are likely to experience more stress. And we know that these early experiences of stress, particularly through stress hormone, but also psychological interpretations of stress, are likely to have a similar an effect on the developing child. And the question sort of stood, and what I wanted to try to answer with some of my own work was, do we see early experiences of stress showing the same patterns as we see as early experiences of adversity on the developing brain? And so here I'm going to show you, instead of a topographic heat map, I'm going to show you a graph. So I'm going to start by showing you lower frequency brain activity. And this study was in about 250 infants. And we had all of these infants were being raised in poverty. And I had maternal appraisals of stress. And this is when babies were 12 months old. And so here on the x-axis, I have stress, so more stresses over here and less stresses over here. And here on the y-axis, we have low frequency power, so that lower brain activity, or slower brain activity. And so if we saw that stress was associated with brain activity in the same way that we saw for example, the early adversity studies I just showed you, we would expect to see that as stress increases, this low frequency power would also increase. And indeed, that's what we saw. So we saw the same pattern here with maternal stress as we had seen in some of those previous studies that I just showed you. What about higher frequency power? So here, again, we would expect the effect to be in the opposite direction, that as stress increases, higher frequency power would decrease. And indeed, that's what we saw. So, a slide has fallen out here, but let me tell you what it says. So, okay, I think I've hopefully showed you across a few different studies now that early experiences of stress and adversity seem to be affecting how functional brain activity develops. Okay, so when we think about that model I showed you earlier, we have identified perhaps an early marker that might be helpful for telling us when early experiences are shaping the brain in a way that may have really serious downstream consequences. But the next question is whether or not this mechanism or this marker is sensitive to intervention. So I'm going to show you two slides that hopefully get to this point. So the first one is from the same study as that first set of heads that I showed you, so the kids who were being raised in institutional rearing. However, the authors went in and they started a high-quality foster care intervention. So they went in, they randomly assigned children to continue getting care as usual by the Romanian government or to be placed in a high-quality foster care home. And they looked to see whether or not this helped or changed how children's brain activity presented itself. And so here, this is what for low frequency power, this is what the care as usual group looked like. So these are kids who largely continued to getting care as usual, which was continued institutionalized care for many of the children. And here's what children's brains looked like who had never experienced any sort of institutional rearing. And here are going to be the two foster care placement groups. Critically, I'm going to show you children who were placed before 24 months of age, so really within that first 1,000 days of life. And here I'm going to show you data for children who were placed after 24 months, so really after that period we're thinking about in the symposium. And what we see is that children who were placed earlier have brain activity very similar to children who had never experienced institutionalized rearing. But children who were placed later did not see this sort of change in brain activity development. So this is a really nice way of showing that maybe these early markers of brain activity are also sensitive to intervention. So in my own work, I've worked on a study where we did a poverty reduction intervention. This study is called the Baby's First Year Study, which is one of the first randomized controlled poverty reduction interventions and the first in the United States. For this study, we recruited 1,000 moms who were living at or below the poverty line when they gave birth to their child. We recruited moms from 12 hospital maternity wards in 4 metropolitan areas. Upon enrollment in our study, moms began receiving an unconditional cash gift that they got every single month. So moms were getting money they could use however they saw fit on a debit card. But critically, we randomized moms into getting different size cash gifts. So we have one group called the high cash gift group. This is 40% of our moms. And those moms receive $333 a month on the debit card to use however they see fit. Whereas our low cash gift group only receives $20 a month on a debit card. Same idea though, they can use the money however they see fit. And the idea here is that we are randomly, because we do random assignment, the only thing that should be different between these two groups is this cash gift that we're giving the moms. This cash gift, again, is trying to alleviate poverty. So what we saw here, I'm going to show you high-frequency brain activity in 2 bands, but I think the graphs sort of speak for themselves, which is that in one of these bands, you can see that the high cash gift group shows more of this high-frequency brain activity in comparison to the low cash gift group, sort of suggesting that our intervention may be impacting brain activity. And we see this pattern also in this even higher frequency band. Now, I will say that this pattern and the study has gotten a lot of attention over the last few years because it is a really promising way of thinking about whether or not brain activity and cash gifts can really change how the brain is developing. So I do just want to say a couple things, which is that, as scientists, we are always very rigorous in where we say we can draw conclusions, or at least we want to be very rigorous in where we say we can draw conclusions. And this pattern was consistent at the whole brain level. We had a slightly truncated sample size. We couldn't measure all of the moms, not because we didn't want to, but because COVID happened, as you might imagine. So the sample I just showed you wasn't all 1,000 moms. It was about, we had 606 in-person visits before the pandemic stopped everything. But it's still a very large sample size for pediatric brain imaging. So I think we should think of that as a strength, even though we didn't get every mom. But I will say those comparisons I just talked about did not survive some of the most stringent tests for multiple comparisons, so that should give us a little bit of caution interpreting in our results. And it does survive some other more lenient corrections for multiple comparisons. So I think sometimes with science, beauty is in the eye of the beholder there. However, when we look at different regions of the brain, some of those regions, specifically in the frontal areas, do survive really stringent corrections for multiple comparisons, but we didn't pre-register those hypotheses. So again, a little bit of beauty in the eye of the beholder. But this leaves us with, where do we go from here? Well, hopefully I've shown you some evidence that functional brain activity may be a really useful marker for both understanding how adversity is shaping the brain, but also thinking about how we measure intervention effectiveness. But so far this evidence is small, and we need much more consistency and replicability, particularly across neuroimaging studies where things like replicability, we know, is rather low, and can be a high incidence of things like p-hacking. We also need to think about how predictive these early markers are of later social and emotional and cognitive skills. We think they are predictive, and there is evidence to suggest they are, but how strong and how tightly coupled those are is still, I think, a question that we need to answer. And I also just want to say that today I talked about one marker, which was EEG. But I will say there are many markers out there that could also be in a talk like this. So when we choose our markers and we think about intervention science, we have to choose the markers that are wisely and think about what those markers mean for mechanism. So for other interventions, brain activity may not be the marker that is right for understanding effectiveness. It may be epigenetics, it may be telomere length, it may be fNIRS. So thinking about how we measure the effectiveness of interventions should really be a mechanistic question in general. So with that, I want to thank you all for your attention and thank my lab, and we'll leave it there.